Charles Darwin brought tangible arguments of the evolution of species, in the idea that a species is different from another species, if by mating they do not have viable offspring, which in turn have offspring. It also showed how the isolated species on the Pacific Islands evolved separately and, depending on the environment and food, their beaks looked a little different. However, no one has been able to prove that there was a link species between one species and another, in the sense of an intermediate species. Because of this, the Society of Scientists has received many reproaches for teaching young people in school a theory that would have fundamental flaws. From current human experience, the idea of evolution is real. As more beings are produced than the useful amount of food required and the ecosystem space required for the species, the living world is one of struggle for existence. The best wins and perpetuates itself. But this is observed within the internal evolution of species. The fact that the same species has different beaks on different isolated islands in the Pacific would not represent so much an evolution as an adaptation to the surrounding environment. Be adaptation evolution? No matter how much a species changes through adaptation, it remains essentially the same. The reality is of a completely different nature. I previously gave an example of the formation of a cell. This is the mirror of the solar electromagnetic spectrum. As the solar spectrum is a long-lasting one, at the structural level the cells have experienced few changes that would lead to their adaptation to the new changes in wave intensities, or the modification of the spectral structure of the solar electromagnetic spectrum. The result of the weak changes in the solar spectrum are the adaptive evolution that led to bacterial, archaeal, and eukaryotic cells, each of them being specific for a long period that created, by changing the solar spectrum, changes at the level of the number of organelles in cells of a new type. Thus, the intensification of visible radiation and solar infrared, as an effect of the cooling of the sun, created through resonance and absorption at the level of atoms, molecules, and macromolecules. The organelles chloroplasts and mitochondria. Chloroplasts resonating and absorbing visible waves, and mitochondria processing and resonating infrared waves. It must be said that in the current literature it is considered that mitochondria were originally separate, self-contained cells. The symbiont theory suggests that mitochondria originated from bacteria that were engulfed by primitive cells. Instead of being digested, the bacteria established a symbiotic relationship with the host cell, becoming mitochondria. Such theories deny the physical laws evident in the structure of living cells and offer a purely biological explanation. The nature of living cells provides a purely physical explanation. The electromagnetic structure and intensity of waves in the solar spectrum are the causes of the structure and slow evolution of living cells. It is practically meaningless to unite two cells on biological aspects. Such a union would mean one at the level of all common organelles. The most interesting aspect of the symbiont theory is the fact that currently there are no more mitochondrial-type cells. They are disappearing in the meantime. For what reasons? We therefore observe that the cell does not evolve but adapts to the conditions imposed by the cellular electromagnetic spectrum, and the species do not have link species between them. There is a simple answer, related to the external photoelectric effect. By changing the wavelength of the incident beam, an electron of a different energy, which means it is of a different wavelength, will be ejected from the surface of a metal. So the photoelectric effect is discontinuous when we change the wavelengths. As I have already reported, the photoelectric effect also happens in the biological environment, only it is not instantaneous. If there are changes in the intensity of wavelengths at the level of the sun, it is as if we are changing the wavelengths absorbed by biological media and resonance in the wave structure phenomenon at the level of structures in the living cell, such as cell organelles. For example, the red radiation emitted by hydrogen has decreased by about 2 times compared to the Carboniferous period, 359 to 299 million years ago, being replaced by the violet radiation emitted by calcium atoms from the sun. During this entire period, the size of trees and land animals decreased drastically. Carboniferous forests were extremely rich and provided food for large reptiles, then dinosaurs. About 66 million years ago, the so-called extinction of the dinosaurs took place. It was concluded that they disappeared following the collision of the Earth with a large celestial body. However, the dinosaurs did not die out suddenly. At the end of their existence, dwarf dinosaurs appeared, and today their relatives are birds. Ostriches, the emu bird, chickens, ducks, eagles, are the direct descendants of the great reptiles. So we could assume that changing the wavelengths at the level of intensity leads to changing the cellular activity. For example, the transition from intense emissions from the red color emitted by the sun and absorbed by chlorophyll, A, to the violet color emitted by calcium made possible the appearance of chlorophyll, B, and other pigments in the chloroplasts of green plants. Such transition phenomena occur periodically on the sun, according to the evolution of the yellow stars in the H.R. Hertzsprung-Russell diagram, as the stars cool. So the change in intensity and importance of wavelengths in the solar spectrum have an evolutionary effect on living matter and especially species. It appears that the extinction of the dinosaurs was accompanied by an increase in iridium, a radioactive element, in soil samples from that time. Some researchers assume that the iridium was of volcanic origin, but the possibility that it was brought in by clouds of matter emitted by solar activity is much more logical. Basically, the sun emits new atoms into space as it manufactures them. It is interesting to note the historical data provided by astrophysics in the emission of yellow stars and the events that have taken place on Earth in geology and the history of life. Fig. No. HR diagram used in astrophysics to elucidate the problem of the evolution of various stars. Authors, Hertzsprung and Russell. Fig. No. The history of intense emissions at the level of yellow stars in the HR diagram is related to the temperature at their surface. This graph, known in contemporary astrophysics, can explain the very evolution of life. The idea suggested by the physical and biological analyses related to the geological activity of the sun is that the evolution of species does not have linked species. The modification of species taking place against the background of modification on other wavelengths of electromagnetic radiation in the solar spectrum. According to the external photoelectric effect, this transition results in the change of wavelengths, a phenomenon that is discontinuous. The effects of discontinuity on living matter being of a completely different nature than evolution. Evolution takes place on a background of changes in the intensity of solar waves, not on a background of appearances or disappearances of intense lines in the solar spectrum. So the transition from one species to another is not done in evolutionary italics, but through discontinuous evolutions with important changes at the level of cells and organisms. These changes are periodically related to solar cycles, which are simultaneous from 11 years to hundreds, thousands, or millions of years. Examples of cycles are periodic glaciations and the growth of annual tree rings, which are directly related to solar activity. In the Carboniferous, the annual tree rings were several centimeters long, and today the growth is of the order of a few millimeters. In both cases, the thickness of the rings was closely related to the solar cycles. The author of this paper performed annual ring measurements on present-day tree slices of spring-summer and autumn-winter wood thickness, resulting in the same connection with the current 11-year solar cycle. The modification of the wavelengths in the solar spectrum, the nearby cosmic environment, but also the terrestrial environment, make it impossible at a given moment for individuals from isolated groups to reproduce, so that a species subject to solar spectral effects separates into two or more species. Isolation is one of the most eloquent events in the evolution of species. Australia is the microcontinent where the life of the species evolved in a completely different way, developing the species left at the time of separation from the other lands, due to the drift of the continents. The evolution that Charles Darwin tells us about, through natural selection, from the emergence of life as a biochemical product, until now, considers life as a given, in which the laws of biological, not physical, needs are important. Here, however, we can bring to light several aspects of living matter dependent on the solar spectrum and physical laws, not chemical ones. The idea that life is a chemical is already meaningless. Spectra, molecular, macromolecular emission absorption of atoms and wave structure resonance are phenomena directly related to solar activity and deeply involved in the physics of living cells. So that life was destined to exist since the formation of the sun and our planet, under the conditions of the existence of an environment of liquid water, hydrogen, oxygen, carbon, and nitrogen. As the external photoelectric effect is instantaneous in metals and at the level of tens of minutes at the cellular level, the building period of the bacterial cell was quite small. Once the cell was created, the first organisms appeared, obviously subject to Darwin's laws of evolution. This type of evolutionism assumes the existence of information and stores of a biological nature transmitted from generation to generation. ADN and ARN are considered to carry this information. In fact, what would be the use of information in biological nature as long as nature behaves impeccably according to its laws? No matter how hard living matter tried to function outside of the laws, it would be impossible. Under these conditions, to exist and evolve, there is no need for transmitted information. One fact is certain. Biological laws cannot alter physical laws in any way. Nothing we learn in life will lead to the evolution of the offspring in terms of retaining the information received, so that each generation has to follow a school in turn. Most likely, the information itself is the huge number of atoms, molecules, and macromolecules in the living cell, each of which has a customized electromagnetic wave absorption spectrum. With the help of these spectra, each cell can be developed separately at the level of intensity and at the level of wavelengths, quantitatively and structurally. This is precisely why the amount of amino acids and nitrogenous bases is fundamental in the cell. To multiply, a bacterial cell will need to double its amount of material. DNA, on the other hand, is a string of nitrogenous bases that individually absorb a large part of the ambient electromagnetic spectrum, bases that also exist in abundance in the cytoplasm of cells. According to the external photoelectric effect, a cellular component that has its own absorption spectrum will multiply if there are waves in the sunlight that feed these spectra. Atoms, molecules, and macromolecules are true absorbers of certain electromagnetic waves, thus preserving the cellular configuration from one bacterial generation to another. The same happens in eukaryotic cells. The number of chloroplasts doubles under the effect of visible light, following sexual multiplication. If we look at a perennial plant, we notice that at the end of the year only the seeds remain, the rest being used as food for other animals or rotting. The seed is the very immortality of any species made up of eukaryotic cells. Does the seed carry information other than the atomic absorption emission spectra of atoms, molecules, macromolecules, and cell organelles?